I have the honor to introduce the first keynote speaker, Professor Peter Bühlman from ETH Zurich, where he is the chair of the maths department. So Peter has done numerous important contributions to statistics and machine learning over the years on subjects like causal inference, high dimensional estimation, semi-parametric inference, Markov processes, and many more. He has such a long list of merits that if I would be here selling out it to the end, he would have no time to give his talk. So therefore, I say to Peter, please tell us about the high dimensional causal inference.
Thank you very much. It's a great honor to be here and to give this opening talk at this wonderful conference. I truly enjoy being here in this environment with these people at the warm beaches here. It's great to be here. So this is again the title of my talk. I'd like to acknowledge my main collaborators in this research, Jonas Peters, Jan Ernest, and Marloes Maathaus. They're all from our seminar for statistics at ETH Zurich. First, I may want to address potential concerns and hopes, namely that I believe we cannot do confirmatory causal inference without randomized intervention experiments. But I'd like to convince you that we can do better than proceeding in a naive way. And this is, in a way, my talk. I'd like to convince you that we can do something substantially better than just giving up. OK, so the grand goals, my main motivation to study the sort of questions I'm interested in are coming from genomics. And the first question could read as follows. If we would make an intervention at a single gene, what would be its effect on a phenotype, on a response variable of interest? And I'm using the subjunctive here because I want to infer or predict these effects without actually doing the intervention. So that would be a great goal or great achievement if a machine could predict what would happen if you make an outside intervention. And my prediction should be based on, say, for this talk, observational data only that are observations from a steady-state system where you haven't actually seen or done the interventions. OK, so if you're working in a different applications, of course, it doesn't need to be genes. And also, we can generalize to interventions at more than one variable or one gene. But for simplicity in this talk, I will focus exclusively or almost exclusively on one variable interventions. OK, so here is a more concrete first example, a project where I have been involved in. It's about flowering of Arabidopsis Thaliana. So here you see that sort of plant. It's a model plant. You can easily grow it. It's a stable, robust plant, so you can do many experiments with that one. And the phenotype or response variable of interest, I denote it by Y, as usual in statistics, is how many days it takes until the plant is flowering, until it is bolting or flowering. And the general aim behind that project is to produce fast flowering, fast growing plants with the general idea of having efficient food production. OK, so this is my response of interest, and I have covariates. I denote them by X, and these are the gene expressions from essentially the whole genome of Arabidopsis. And there are a bit more than 20,000 genes and their expressions involved. And the question is, can we infer or predict the effect of knocking out or knocking down a single gene on my response variable of interest, my Y, which is how many days it takes until the plant is flowering? So you want to kind of cleverly genetically engineer new mutant plants. OK, so here is a multivariate version of this first example. And it's again about gene expressions. It's in yeast. This is baker's yeast, which you use for making your bread in the morning. So we measure again gene expressions. We have a bit more than 5,000 genes involved here. And it's a multivariate version of the first example. I have a first response variable Y, which is the expression, say, of the first gene. And then I have many covariates X, which are the expressions from all other genes. Then you can go to another phenotype or response variable of interest Y. This is the expression of the second gene. The covariates are the expressions of all other genes, and so on and so forth. So in total, you have 5,360 response variables, and all other variables are the covariates. And the question is, again, can we infer or predict the effects of a single gene knockdown on all other genes? That's the question. Obviously, we consider here the framework of an intervention effect. This is sometimes called the causal effect. In my opinion, causal is a very, very ambitious word, and intervention comes a bit closer to what we're actually after there. So we want to predict something on an outside intervention, what is its effect on a final response of interest. Okay, so here is a slide which should convince you that regression, which is, I think, the statistical workhorse in many applications, is fundamentally the wrong approach. Think about this Arabidopsis example again. So this is kind of a classical, at least statistical data set. You have a response variable y, you have covariance x. Suppose the y and x would be even jointly Gaussian, then this is the correct model. You can always decompose y as a linear function of the x's plus error term in the Gaussian case. In a non-Gaussian case, you can declare this is my model. So this is what you often would do. You would relate y as a linear combination of the x's plus error. Suppose you're willing to scale your covariates, say that they have variance 1 or empirical variance 1. And then I think we all would agree that the regression coefficient beta j, or its absolute value, it measures the effect of xj on y in terms of association. And what does association mean? This is very trivial, of course, but very important to keep in mind. It's the change of y as a function of xj when keeping all other xk's fixed. So this is textbook, of course. If you change xj by 1 unit, beta j measures that change when you keep the others fixed. Okay. Now the question is whether this is realistic, and I claim this is in many applications not very realistic. If I do an intervention at a variable xj, why should the others be kept fixed? They cannot be kept fixed. They probably change as well. If I do something, an intervention at my jth gene, maybe many other genes will change as well, and I cannot keep their values fixed. So this is my explanation why regression is fundamentally the wrong approach to capture the effect of an intervention. And indeed, it's not only, fundamentally or methodologically the wrong thing, it's also from an empirical point of view, really the wrong thing. So here's an example. This is a real system. I'll explain in a minute what it is. But it is some sort of an ROC-type plot. On the x-axis, you have false positives. On the y-axis, you have true positives. And the true and false positives are with respect to an intervention that has been made. So the blue dotted line here is random guessing. Then you see 2 lines, the red one and the light blue one. They are regression methods. The red one is the lasso. The blue one is the elastic net. And there's a black one which does substantially better than the regression methods. And what I want to convince you is that indeed, with this black curve here, we can do much better, substantially better than modern state-of-the-art penalized regression. And it's not a problem that these regression tools are not good. The regression tools are the wrong tools for answering the sort of question I'm interested in. The black line is something which is based on causal inference modeling, on graphical modeling, and I'll explain a bit more during the talk. So what is the example about? It's my yeast example. It's the effects of single gene knockdowns on all other genes. I remind you again, we have more than 5,000 gene expressions, all genes from the yeast genome. We measure their expressions, and we also have 231 gene knockdowns. So there were 231 single deletion strains of yeast. And you can see what happens. So you knock out the gene in yeast, and you see what is its effect on all other genes. And then you see some huge effects. If the effect is huge, you declare it a true positive. And if it's not that large, you don't declare it a positive. And then, thanks to these intervention experiments, you're able to draw an ROC-type plot, right? You can say what was the truth, what is not a truth, and you can see how well you can predict the truth. The truth is predicted based on observational data only. So there's 63 wild-type yeasts with no knockouts, and you just measure their expression, and you try to infer from these observational data the intervention effects which you actually have. So this is kind of the test set, the 231 intervention experiments. They serve as a test set, and I'm simply looking how well can I predict these 231 experiments. And here is again the performance. I'm not claiming that this black thing is a great performance. I'm just claiming it's substantially better than random guessing, and it's substantially better than regression techniques. Okay, so let me explain a bit what is behind this machinery with the black line. And I'll try to introduce a bit the concepts. I'll talk about DAGs, structural equation models, and causal effects. So for simplicity, let's look at a univariate response Y. Like in my Arabidopsis example, Y is the days it takes until the plant is flowering. And I have, say, a p-dimensional covariate X. I have other variables which I measure. And the question is, what is the effect of setting the jth component of X to a certain value, little x? And I formalize this using Pearl's do operator notation. I write do xj equals little x. Obviously, this is a question of intervention type, and it's not the regression effect of xj when on y when keeping all other xk's fixed. So it's this kind of dynamic intervention effect. Okay, how can we capture that dynamic notion? And graphs are very useful for that. At the beginning here, I already showed the two main assumptions we place, at least for this exposition, for this talk. One assumption is that there is an underlying true causal mechanism, and I assume that the causal mechanism can be represented in terms of a DAG, a directed acyclic graph. In particular, I'm not allowing directed cycles, so that's forbidden. There are relaxations of that sort of main assumption. Here are references for that. It's an interesting topic, but I will not address it here. And the other thing is, I also assume there are no hidden variables in my analysis. So if you have hidden variables, hidden confounders, it becomes a very tricky, different problem. People are working on that as well. This is an interesting area as well, but I will not talk about this today. So maybe in my genomics applications, I measure many variables. And that's in a way a blessing with respect to the hidden variable problem. I hope to have measured all relevant variables. Of course, it's not quite true, but maybe it's a good approximation. While it is a blessing for that part, it is, of course, a curse in terms of estimation. If you have many variables, you have more problems when it comes to statistical estimation. Okay, so let's work with these two assumptions. And now let's look at structural equation models, which are quite simple, I think. So I consider p variables, x1 to xp, and should think y, my response variable, is one of the xj's. And then I write a structural equation model in this form. So xj, I don't write an equality. I write this arrow from right to left. It says xj is generated by a function fj, and I have the parental variables and an error term epsilon j. The epsilons are IID error terms. And what it means here is that only the variables which correspond to the parents of the jth node, the jth variable, play a role in this equation. So here is an example where you see how this goes in action. So here is my y variable, and the structural equation could look as follows. So x3 and x4 are root nodes, so up to transformations, they're just error terms, epsilon 3, epsilon 4. x2 can be almost an arbitrary function f2 of x3, x4, comma, error term. The error doesn't have to be additive at the moment. x1 is a function of x2 and an error term, and y is a function of x1, x3, comma, error term. Again, the errors do not have to be additive yet in this model. Okay, so this is a general structural equation model, and now let's talk about how can we capture the issue of an intervention distribution. I make an intervention at a variable x, and I write do x equals little x. Okay, so what is the idea? The idea is very simple. You can go as follows. In your structural equation model, you set the corresponding variable large x to this value little x, like 2.7, and then you propagate the effect according to the structure of the model. Now, there's one additional important assumption here, which is used by almost everybody in the causal inference literature. The assumption says the structural equations do not change under the intervention. If I make that intervention, the equations remain the same. That's a major assumption as well, but it's a convenient assumption. Okay, so what do I mean by that? Let's look at the example here. I make an intervention at the second variable, so I write do x2 equals little x, and now I propagate that effect. This is very simple to do. If I set x2 to little x, These things do not play any role, right? It's obvious. So x1 becomes this function f1 of little x. I set x to the little x, comma epsilon 1. And y is a function fy of x1, x3, comma epsilon y. This is the definition. Now you plug in your x1 here, so it's kind of an iterative procedure. Now x1 is f1 little x, comma epsilon 1, comma x3, comma epsilon 1. And as you see, it's quite a complicated expression already. But that's, in a way, the random variable y when having done the intervention at the second variable. Now, you're interested maybe in the distribution of that y when doing the intervention at x2 equals little x. And in principle, it's a complicated formula. And in principle, you could address this, say, this Monte Carlo approximation. If you know your functions f, if you know the error distributions, you can at least approximately compute the distribution of y when doing the intervention at the second variable. But it looks kind of complicated, and it is. OK, so here is a great trick, and I think it's mainly used or only due to Perl. And Perl says, OK, you don't have to go through this complicated exercise and propagate the errors. You can do some sort of adjustment. And Perl at least showed it in a very convenient, tractable form. So here is actually the distribution of y when doing the intervention at variable x and setting it to little x. And there's a formula. And the formula is called the Backdoor criterion or adjustment. And I use it here in connection with the parental sets. So if I do an intervention at x, I look at the parents of x. If y is not in the parental set, if it is, it's uninteresting. If y is in the parental set, there is no causal effect from x to y. So the only interesting case is if y is not in the parental set. So if y is not in the parental set, I can get that distribution, not through this complicated form. I can get that distribution by simply, I take the standard conditional distribution of y given x, and say the parental variables, and then I need to integrate out over the parental variables. Okay. And I can, for example, also get the expected value of my variable of interest, y, do x is little x. Now you just use the definition of an expected value. It's just the integral y, py, do x, little x, dy, right? So it's very simple. All what you need is the parental sets of the variable where you do the intervention. Now here comes an important additional remark. You can actually take any other set than the parents. You can take a larger set, for example, than the parents. I call it Sx. It must contain the parents, and Sx should not contain the descendants of x. That works as well. Then you replace here the parental set with this Sx. For example, you have a DAG. Say it's the true DAG, and you go to a larger DAG. You put additional arrows in your DAG. That's called a super DAG, and you could take the parents from that super DAG. So Sx could be the parental set of a super DAG of the true DAG, and the things here work as well. Okay? So for the intervention distribution, it's actually sufficient to know only the superset DAG, which contains the true DAG. That's sufficient. And I need the observational distribution of that triplet y, x, and the variables in this Sx. So I go into this formula. If I have that observational distribution, I'm done. That's great news. And let's look in the Gaussian linear case. There it becomes particularly easy and explicit. Suppose my variables are jointly Gaussian. Then you can actually compute that the expected value of y doing an intervention at x is a linear function in this little x. So it's linear in little x. This is not the conditional expectation, but nevertheless, this is linear in little x. So if you're interested at the intervention effect, what people typically do is you take the derivative of this quantity with respect to little x. If you do that, it's a constant, right? It's this theta here in this linear function. And so the intervention effect is actually, in the Gaussian case, a parameter. So this is a parameter, and this is sometimes also called the causal effect. So it's a parameter, and how can I get to that parameter? Again, Perl's backdoor criterion says if y is not in the parental set, I can get that parameter through regression. I run a linear regression of y versus x. This is where I do the intervention, and I take all parents of x into my model, and the corresponding regression coefficient for the variable x. This is the causal effect. This is the intervention effect. So you only need parental sets or parental supersets and linear regression, and you're done. Okay, so what is that? So when having no unmeasured confounder, this is my assumption, the intervention effect, this is the typical classical or a-classical definition. This is called a causal effect. So this ambitious word, causal effect, is now linked to a clear mathematical principle. This is the causal effect. And what is the causal effect? This is the effect from a randomized trial under the made assumptions. But of course, you don't want to run this randomized trial because it could be very expensive, it could be unethical, it could be impossible to do. Okay, so that's the setting of the scene. Okay, so this is the causal effect, the intervention effect. Now we should ask the question, how can we infer these intervention effects from the observational distribution? And I'm looking at different regimes. I first look at a simple scenario, which I find quite useful actually to look at. Let's assume the DAG is given, or a superset of the DAG, or maybe more generally, the order of the variables is given. Okay? So then we can say something, and what we actually only need to do is sparse regression. The second scenario is the DAG is unknown, and then I have two bullets here. I have the fully identifiable case, so we can learn the DAG from the data and then proceed with that first point here, or the partially identifiable case, then we cannot learn the DAG, we can only learn an equivalence class of DAGs, and we only can get lower bounds of causal effects. I will not talk much about this today, and there has been a lot of work on identifiability, what you can do and we cannot do. So let's first look at the simple problem where identifiability is not an issue at all. Suppose somebody gives you the order of the variables. Maybe in a time series setting, you would also always propagate the order from the past to the future. Okay? And now let's look at a very general structural equation model. Xj is generated by any function fj depends on the parental set, comma, an error term. The error doesn't have to be additive. The only thing I require is that the errors are independent. Okay, so if somebody gives you the order of the variables, then you would know the parents must be the previous variables. Not all of them, but some of them. Suppose you take all of them. I'm interested in xj as a function of the previous variables. So in principle, you could hope to address this issue with sparse regression. You do a regression of xj versus the previous variables. But of course, this is very difficult in the general form. If fj is some sort of multiplicative form, if the parental set is very large, and so on and so forth, you run into the curse of dimensionality, and you run really into the curse or the problem of model misspecification. Maybe you have worked with the wrong model here. I think almost nobody tries to address this problem in full generality where fj is just any function of the parental variables and the error term. Okay, so this seems very ambitious, but here is a new approach which is actually very simple to solve at least an aspect of this problem. And I show you the result in terms of a theorem. So I take a general nonlinear structural equation model with essentially any functions fj. They need to be in L2P, and I need a bit of regularity conditions, but just this is minor, and you can talk with me after the talk about that. And now what I'm interested in is not the general structural equation model and its parameters. I'm only interested, say, in this quantity, the expected value of y do x is little x. This is my stuff which I want to infer, say, in biology. Obviously, this is a quantity, and this is a function g of little x. This depends only, the left-hand side depends on little x. So it must be some function g of little x. And the issue is now, and this is, I think, quite beautiful, this g of x has a very explicit characterization. This g of x is simply the best additive L2 approximation of y versus x, and say the parents of x, or a superset of the parents of x. Again, so this is some function g of x, and I have a very explicit characterization of that function. It's simply an additive regression, a best additive regression function when I run an additive approximation of y versus x, and the parents of x, or maybe a superset of the parents. So here's the mathematical formalization. Here's the quantity I'm interested in. G of x is the best approximation. What I mean by that is here the argmin. I have expected value. I have the L2 error here. I look at the best approximation. And then the quantity for the variable x here is, in fact, this function g of x. So this is a very clean result, I think. Of course, we need to know the order of the variables, or the parental set, or a superset of the parents. But the issue is, I only need to run additive model fitting, even if the SEM, if the structural equation model is super non-additive, is very general, is very multiplicative, or whatever. I only need L2P functions. So that means, and I call it actually the ORD additive model fitting, ORD for order. If I have the order, I just fit an additive model with respect to that order. Say y versus x and the previous values of x. I call it ORD additive modeling, ORD additive model fitting. And the issue is this is very robust against model misspecification. It's hard to come up with a model where this is wrong. It's just simply right. I have some sort of small regularity conditions. I give you some sort of a clue how you could be wrong with that model. One misspecification could be due to dependent epsilons. So if you have dependent epsilons, that would mean hidden variables in your problem, and I have excluded that. So this is not robust against the hidden variable problem, of course. Also, here you see a bit why is this working? So suppose you would be interested in the expected value of y and you do a two-variable intervention. I'm not talking about that afterwards anymore. If you would do a two-variable intervention, I would actually need to fit a first-order interaction model. So it's only because I use a single-variable intervention I can go away with an additive model approximation. Okay, that's in a way the trick. If you're only looking at single-variable interventions, additive modeling is sufficient. Okay, so you came all here to Reykjavik. Most of you traveled here, and you bought an airline ticket. I bought one as well. I flew actually with Swiss, but it was a Star Alliance flight. So this is a project with Swiss, and you as a customer, you want a cheap ticket, and of course the airline wants to maximize the revenue here. So here's the revenue, and you can try to maximize that by clever pricing strategy. And this is the sort of cartoon what these people have. It's actually a very complicated problem, but the sort of cartoon is, okay, if you change your pricing strategy, what would be its effect on the revenue? And then you try to maximize the revenue. And here's a kind of cartoon. There's many other variables in the game. Of course, it's some sort of confidential what kind of variables they use. But you have lots of other variables. Maybe you have an order of the variables. They actually have the order. Of course, it's wrong, but they use that sort of order. They have lots of other variables. They draw DAGs. And then all what I'm saying is, okay, if you want to know the expected value of revenue, doing an intervention at pricing, all you need to do is to do an additive model fitting. It's robust. I'm not talking about any sort of complicated nonlinearities and so on and so forth. It just works. That's the beauty of this method. So in practice, if you do estimation, this is easy, of course. You run an additive model of y versus x and say the parents of x or a superset of the parents. In statistics, I think Hasty Tipshirani, Wood, there are many books on that topic. So my talk is also on high-dimensional causal inference. That's easy in that case as well. Because, so you now run, say, Sx is large. Suppose I do an intervention at x and I take all previous values. That's my Sx. So if that is large, I'm in a high-dimensional setting. But that's easy to deal with. So even if the Sx is 100 or 5,000 and sample size in the 50s, we know all the results on additive modeling which have been worked out. So consistency, optimal convergence rates for sparse additive model fitting. So if the problem is sparse, you need to enforce that, otherwise you have a problem. And in our case, this means if the true DAG and its maximal degree is not too big. So if your maximal degree of the DAG is not too big, that is, if the DAG is sparse, you are in a sparse regime, you need an identifiability result or condition, which is kind of complicated. I'm not explaining it here, but it's well established in the high-dimensional inference literature. It's a restricted eigenvalue condition. Then you can do high-dimensional additive fitting, and it applies here as well. So all what I'm saying is additive model fitting is really well established. It works I think, quite well, including the high-dimensional setting. So this is all when the order of the variables are given. So here's an example. This is a pathway in Arabidopsis. I've shown you the Arabidopsis example before. That's a bit a different problem. That's an easy problem in a way. So this is a pathway which produces the chlorophyll, which is needed so that the plant can green. And I have 14 variables in the game, 14 genes in this pathway. I have 118 samples. And I assume the orders of the variables are given. How do I do that? I just take biological information with respect to up-downstream. That's, of course, questionable whether you want to do that or not. But suppose you just go with the up-downstream information from biology and you declare this is the right order for your causal inference. And you can do that, and that's actually indicated here in this graph. So the black arrows just indicate the order. So this gene comes first, and then this, and so on and so forth. Okay, now you just run additive, or this ORD additive modeling with that order. And I'm interested in scoring gene pairs X causes Y in terms of the estimated intervention effect on Y when doing an intervention at X. I score, I look at the difference, I subtract the global mean, that takes square error, and this is my score. I measure how big is this quantity. So the bigger, the better, the more effect. I can take the top 10 scoring directed gene pairs that X causes Y, and then I do some sort of subsampling exercise, which I find quite useful in practice, so maybe you're not sure whether your findings are stable. So you subsample your data, you do it again and again and again, and you see how many times have your gene pairs scored at the top. And so there's actually a formula which allows to quantify the false positives. So if you allow expected number of false positives less or equal to 1, the stable gene pairs or relations are the red ones. So there are two genes here, this one and this, and they're kind of mostly influenced by others, and here are the others which kind of give a large intervention effect on these two genes. This is not validated at all. This is some sort of more a toy problem where you see how this goes in action. The issue is, again, I don't need to worry about complicated nonlinearities or non-additivities. It's just the right thing if the order of the variables is correct and if I don't have hidden variables. Okay, so this ORD additive regression is a local procedure, it's a local operation, and this is actually quite useful in practice, I think. So in principle, you have two ways how you can infer the expected value of Y do X is little x when, say, the DAG or the order is known. One of the procedures is what I just explained, the ORD additive regression. You run an additive regression of Y versus X and the parents of X, or a subset of the parents. Okay? The other thing is maybe the classical thing. If somebody gives you the DAG, what you can do is you collect all directed paths from X to Y, right? You collect them all, and you multiply the edge weights or the edge functions, you integrate all together, and you can also compute this quantity. The question is, which one is the better one? And from a theory point of view, we don't have an answer, but from an empirical point of view, we have a quite convincing story, I think. So here is a setting with a non-sparse DAG. Here is a setting where a true DAG is sparse. What you see on the x-axis is kind of the contamination of the true DAG. On the left here, you have the perfect knowledge of the DAG. So here the DAG is perfectly known. And on the right here, the DAG is not known. It's imperfectly known. For example, on the right here, only 34% of the edges are not correct. So this is a perturbed DAG here and there. So only 34% of the edges are incorrect, and the remaining 66% are correct. Okay, a third of the edges are incorrect. Now, if you have imperfect knowledge of the DAG, and I think that's quite realistic, if you estimate the DAG, that estimate will not be perfect. Then this ORD additive regression, that's actually the box plot on the right-hand side, the dark one. And this is the squared error. So this box has better performance, shows better performance. And on the left-hand side, you see this method where you integrate over all directed path. Obviously, if the DAG is incorrect, integration over all the path must produce something quite wrong because you integrate up over many wrong directed paths, because it's a very global operation. Now, if the DAG is perfectly known, it's the other way around. If the DAG is perfectly known, this method actually makes use of the additional information how the DAG looks like. It takes into account the whole global structure of the DAG, and it's a bit more efficient here, you see it, than the order additive regression, which is a bit less efficient in terms of statistical performance. Okay, so from a more practical point of view, the order additive regression is much more reliable and robust when having only imperfect knowledge of the DAG, and I think that's quite realistic in practice. If you estimate the DAG, or if somebody says, this is the true DAG, you should never believe that guy. It will be imperfect. So I think the order additive regression is much more robust. Okay. So let's move a bit into what can we do if we don't know the order of the variables. And I'm only looking at the fully identifiable case here for simplicity in this exposition. I listed 3 important identifiable structural equation models where the DAG can actually be identified from the joint observational distribution. Somebody just gives you the distribution of x1 to xp, and then you can identify the structure of the structural equation model and then the parameters. One nice example is Lingam, proposed and advocated by Shimizu. Here he is. So this is a linear structural equation model, but with non-Gaussian errors. And at that time, I found it, I think it's a beautiful contribution. And the question is this identifiable? And the answer is yes, it is. It's the same story as with independent component analysis. The non-Gaussian components are identifiable. Identifiability for Gaussian components is ill-posed, and you cannot identify. So thanks to the non-Gaussian errors, this is identifiable. You can also look at linear structural equation models, which are Gaussian. In general, they're not identifiable, but if you restrict the error variances to have all the same error variance, then it becomes identifiable as well. And here is another nice example where we actually use this sort of result from Bernhard Schellkopf and all his co-workers. This is a nonlinear structural equation model with additive noise. So here's my structural equation model, and it's important to have here this additive noise structure, and the fj must be a nonlinear function. So then again, the structure is identifiable from the joint distribution. Why does that work? I learned that from Bernhard and his former student, Jonas Peters. Let's look at the toy example with 2 variables. So I have a DAG. The true DAG is x1 causes x2. I denote x1 by x, x2 is my y. Suppose the truth is y is x to the power 3, say, plus epsilon. I take Gaussian epsilons, and the epsilons are independent of x. So I have here the scatter plot. Here is y, here is x. You see the nonlinear true function in red, and you see the uniform scattering of the points around the true function. So here you see the noise. Now you try to do that in the reverse thing. You try to represent x as maybe a nonlinear function of y plus error term eta. So you revert the picture here. X is y to the 1/3 plus some sort of error term. And now the more curvature you have, the more heterogeneity of the errors. So what you see is if you go into the reverse mode, the errors are not homoscedastic anymore, they're heteroscedastic, and that means they must depend on the regressor y, and that's forbidden. So the model says, the structural equation model, the errors must be independent from the parents, and so this is not the case, so you cannot write this distribution in the reverse mode, hence it is identifiable. It's a beautiful trick, a beautiful contribution. Okay, so this is quite well known in general. Say if you look at a Gaussian structural equation model or a very non-parametric structural equation model, it's not identifiable. So this is the easiest case, a linear Gaussian structural equation model. It cannot be identified from the distribution alone. You can only identify the corresponding Markov equivalence class. I'm not going into details about that. So what is the strategy now mainly for the identifiable models? How can we estimate these parameters and the order of the variables or the structure of the model from data, from the observational distribution? And the strategy is kind of, I give like 2 modules, and I find them quite useful, and this works actually quite well in practice. First, order search, the order among the variables. Once you have it, you can go to the first part of my talk. Then you just need to do sparse regression. Okay. So order search is, of course, an extremely hard problem, and I show you a bit how we address that issue. So what we do is we do maximum likelihood for the best order or the best permutation. So I have a candidate order or a permutation pi, which is from the numbers 1 to p, a permutation. And if you have an order, you can run a regression. So if you have ordered your variables and the order is now indicated with the pi, I run a regression of x pi j versus the previous values in that order. Okay? So how can you evaluate the goodness of that order? Well, take the likelihood. Now, in this structural equation model, the likelihood is multiplicative, so the likelihood can always be written as the product of the densities or the distribution of these random variables x pi j versus the previous x's and multiply, and that gives me the likelihood. So this is, in a nutshell, what we're trying to do or what we will do. Now, what you need, of course, is a model for these regressions, the functional forms, for example, additive, linear, an assumption on the error terms, Gaussian or maybe non-parametric, and then you can compute the likelihood with the estimated parameters, and then you search for the best order. Okay, so that's a pretty ambitious program, searching for the best order, searching for the best permutation because there are p factorial permutations there. And here comes a trick which is very useful from a theoretical perspective, and also empirically we find this quite important. What we do is, or advocate is, we do a preliminary neighborhood selection. I'm aiming for, in a first step, an undirected graph which contains the skeleton or a larger set than the skeleton of the true DAG, or in other words also, I'm going for the Markov blanket or a superset thereof. So I have an object like this one. It's an undirected graph. It's some sort of associations between the variables, and its meaning is that this is a superset of the skeleton of the true underlying DAG. So it's a superset skeleton, and once you have that, you restrict everything, of course, to this reduced search space. This is an obvious idea, right? So now you do order search and estimation restricted to that superset skeleton. It's much reduced search space, and the other very convenient issue is you can do unpenalized maximum likelihood estimation in this case. You don't need to regularize any further. You just have regularized somehow here with this superset skeleton, and then you're done. So now you need to implement that idea a bit more concretely. So we cannot do that in general, because we need a model for the regressions, and here is kind of our model which we propagate and which we find quite useful. We say, okay, let's look at an additive structural equation model. So here is a nonlinear structural equation model, but the functions of the parents are additive functions. Why? Well, this is still an identifiable structural equation model. It's statistically feasible because I have additive functions. They're quite well behaved. They're not too hard to estimate, and it shows good empirical performance. So here's again how we proceed with that. We do this preliminary neighborhood selection. I get this superset skeleton in a first step. This is my step 1 here. And how you do that, you can give conditions such that the following is sufficient. You just run an additive regression of xj versus all variables. It's very easy to do. You need to make one identifiability condition, but you have that one, you can ensure that you do actually get a superset skeleton. Once you have that, you do unpenalized MLE restricted to the superset skeleton. So what happens is here you get arrow directions on your undirected graph. And if you're interested in edge pruning, at the end you can run again penalized additive modeling, penalized regression to prune away some of the edges. So this step 1 and step 3, this and this one is very well established. In the CAM case with additive models, you only need to run sparse additive modeling. And the harder issue is the point 2. How do we address that one? Well, we do greedy search on the restricted superset skeleton. We don't know how to deal with that in general, but the search space is much, much smaller, so greedy search is quite good. If it is, for example, a tree, we can have an exhaustive computation, and under some conditions, these people have shown that greedy search methods actually give you the exhaustive optimum. Okay, so maybe one slide on the theory. Does that work? Well, under some conditions, let's look at a case where you have very many variables in your structural equation model, p variables, and sample size is only n, is of much smaller order of magnitude. What are the main assumptions such that you succeed with this method? The first one is always you need sparsity. You cannot do high-dimensional inference without a sparsity assumption or some sort of smoothness assumption. We do it in sparsity. And here it reads that the maximal degree of the true underlying DAG is sparse. The second assumption is a tricky one, and it's a very non-explicit one. You need to have such that the model, the true model, is sufficiently identifiable. And identifiable means here with respect to the Kullback-Leibler divergence, because we do maximum likelihood. So you need to have a sufficient gap between the true model and all wrong permutations, and there's some sort of a gap parameter, and that must be sufficiently large. Then you need some moment conditions, and then you can prove that the MLE, the unpenalized MLE on the reduced search space, actually gets a true permutation with high probability, and the probability converges quickly to 1 as sample size increases. With that, if you have an order, you're back to part 1 of my talk. Okay. So I don't have much time left. I think it's nice to end my exposition with what can we actually do in terms of biology. Prediction of intervention effects in biology. This is the first example I've shown you already. We actually used the older technique on linear Gaussian structural equation models and bounds on causal effects with IDA. This is not this nonlinear method I explained today. But at least this shows that there is potential to do something with that. Now here's the modern version of this example. It's the same setting. It's more data, it's more modern technology. This is in collaboration with Frank Holstege from Utrecht. This is still work in progress. It's again about single deletion strains in yeast. I have 160 observational data points. I have more than 6,000 gene expressions, and I want to predict what is the expected value of the expression of gene J when I do an intervention at gene K. I have 300 interventional data points to test my predictions, and here are again these ROC type plots. This is a zoom in of that one, so let's look at this. This is random guessing, and here are a couple other methods. The black one is the CAM method. It's the nonlinear structural equation model with additive functions. And the red one is our old method, the IDA, based on a linear structural equation model with Gaussian errors, which is non-identifiable, and you need to go with bounds of causal effects to rank. So the conclusion so far in this work in progress is we can again do substantially better in random guessing, and it seems like that CAM, this nonlinear structural equation modeling technique, has a slight edge over the more classical Gaussian linear structural equation model. Conclusions. With known order of variables, if you're only interested in expected value of y, do x at little x, you can do this order additive regression. And that's very easy to do. It's very robust against model misspecification. If you don't have the order of the variables, the focus today was on nonlinear identifiable structural equation models, building on the work from Bernhard and his co-workers. We propagate here or advocate order search first and then sparse regression. We have here this trick with the preliminary neighborhood selection, and that actually gives good theoretical guarantees and interesting empirical results. I would like to spell out a warning at the end. I think mainly in the context if you have unknown order of the variables. You have so many uncheckable subtle conditions. And I think you should not claim that your causal inference is confirmatory. But I think it's potentially very useful for prioritization of the next experiment. Say in biology, what should be the next gene knockout? Experimental validations are very important in this whole field of causal inference, and simple organisms in biology are ideal for pursuing this task. Thank you.
Thanks, Peter, for an inspiring, wonderful talk. Now it's time for some questions.
Hi. I had a question on the setup of the problem. at the very beginning, we're looking at the expectation of y conditioned on an intervention on x. Wouldn't we want to condition on observations of the other x's that weren't intervened on before the intervention, or?
Good point. So I think what you're mentioning is a bit closer to this Granger causality thing, for example, where you do conditional on other variables. I think in some areas, in some contexts, this is indeed what you want, and it's much more complicated, except when you go for Granger causality, where everything is linear and the order of the variables is known, then you get much closer to, I think, what you want to ask with your question. So in principle, I could do that, but in terms of estimation, I have to condition on the others, and makes it very hard.
So here we're marginalizing out over everything that we're not intervening on?
Exactly.
Thank you.
More questions?
I was wondering, do you assume faithfulness in the methods that you talk about, or you're able to not assume like the faithful assumption for the DAG?
So let me just briefly think what the correct answer is. So the faithfulness assumption, of course, is needed for, it's typically needed much more in the case of unidentifiable models, where you go for the Markov equivalence class. So here, you also need it in the form that you don't have some sort of cancellation. So, but it's, I think and believe, much less an issue in identifiable models. So you need the causal sufficiency, I think it's called, or that you don't have zero edge weights and so on. Of course, you need that sort of assumption. But in my view, faithfulness is, we need it implicitly as well, but it's kind of obviously implied, usually, under the identifiability assumptions. And it's much less an issue than when you go with these non-identifiable models.
I have a question myself about so in gene expressions, typically the measurements are a sample of a stochastic process or a dynamic system where you actually may have some feedback between the different genes. So they can turn each other on and off. So then obviously in some cases the data actually don't follow any DAG. But I would believe that under certain assumptions your approach is still robust to that. But do you think it would be possible to improve upon that if one would approximate a system where you actually allow for feedbacks?
I agree, it's possible. I don't know how, or possible. So if you look at these sort of curves, now we're here, maybe somebody can come up with a curve like this. I don't know how to do that. Of course, this is again, these model assumptions, they're very probably crude approximations of the truth. We talked a bit before, if you make your model more flexible, that's of course better if you allow for, say, directed cycles. On the other hand, you have a model complexity and it's not clear with finitely many data which of the methods will win. Yeah. But I think it's an interesting, also conceptual problem, and people are working on this.
Yeah. I was thinking that a pseudo-likelihood approach might actually be computationally scalable to solve the problem when you have this loopy structure there.
It becomes much more complicated, right?
Yeah, it becomes, but I think it could still be computationally doable with pseudo-likelihood. Okay, okay.
Why is the random DAG doing so well there? That's kind of surprising.
So this is still very preliminary. So, but what you see actually here, and I'm not, this is in a way a bit too early, is was actually, so it's the blue one here, which is it's in the middle, right? This was actually taking a random order or a random DAG. And here you see also maybe the pitfalls yet in this analysis. So this should be really interpreted as some sort of preliminary work. It's a the problem is, if you look a bit closer to what these results actually produce, for example, this one, which seems to be a success, and I think it still is a success, but if you actually look how the underlying estimated Markov equivalence class looks like and how stable it is, it's a disaster. So I'm very, I mean, try to be honest on that. It's just a very hard problem with so many variables and so few samples to have very stable estimates of your graph or your Markov equivalence class. But if you run this, what I always propagate in any practical application, for example, you do this stability selection, you do some sort of random subsampling. The graphs are terrible, but some of the causal effects are stable. And so there is this thing maybe going back to this one. So even with imperfect knowledge of the DAG, some of the methods might be much more robust. But it's a good point. So at that point in this preliminary work, the random DAG is a random order. I don't know whether this has relations with, say, random projections or anything like that, but somehow random orders aren't sometimes not that bad, which is a bit disappointing and encouraging. So.
Okay, so if there are more questions, so please approach Peter during one of the breaks, and let's thank him once more.